All right, so this is part 4 of chapter 43. This is the way that antibodies work to eliminate pathogens. So the first one is just called neutralization. Here we have a viral particle, and on its surface are antigens. Now, these antibodies are very specific for this specific antigen. So they bind to it, the variable region binds to the antigen, and neutralizes it. And this is important specifically with viruses, because viruses use these antigens to dock with host cell surface proteins to then trigger the host to allow the entire viral particle into the host, or to allow injection of the viral DNA into the host. And because the antibodies are bound to the antigens, this virus now is prevented from binding to a host cell. Now what you'll basically have is in the bloodstream or in the tissue fluid, you'll have viral particles floating around with antibodies attached, and you'll have macrophages and various other phagocytic cells consume this entire thing and take it apart. Agglutination essentially is a science word for clotting. And here we have an antibody that agglutinates or clots, much like blood clots, where you have all the bacteria are attached and kept close by or close together in one small area. Now, what I want you to think of in this way is I want you to think of Pac-Man. Pretend you're playing Pac-Man, the hardest level ever, and part of your job in Pac-Man to score points, I should say Mrs. Pac-Man, because regular Pac-Man is boring. It's too slow. Miss Pac-Man is much better. When you're playing Mrs. Pac-Man, you're trying to eat all the ghosts. So you eat a pellet, and all the ghosts turn purple, I think, and they're able to be eaten. So you start going around and eating them. But imagine if there was a special antibody that took all those ghosts and collected them together, so in one fell swoop, you could eat all the ghosts and get all the points right at the same time, right at once. And immediately you could score, gain the high score and be the best Mrs. Pac-Man ever. This is essentially how these antibodies are working. They clump all these bacteria together in one small area so that macrophages, instead of going around all over the body trying to look for individual bacteria and eating them one at a time, it can take one big bite, if you like, and eat all of these at the same time and be far more efficient at eliminating the bacteria. Hopefully that weird metaphor made sense. Precipitation. To precipitate means to take out of solution. So these bacteria here, these antigens, are soluble in solution, which makes them slightly more difficult to find. They're a bit more mobile. So these antigens, when bound, they precipitate them or cause them to come out of solution, and thus make it easier for phagocytes, like macrophages, to find them and consume them, to eliminate them. Now the last one here is the most complex one, which we'll actually talk about the least, where these receptors, these antibodies, when bound to the antigens, they can recruit complement proteins to the cells, targeting them for destruction. So here you see them recruiting the complement proteins, triggering the formation of a pore and causing cell lysis, causing apoptosis. So what you need to know is antibodies can recruit the protein complement system to trigger cell death. Now, you heard me talk previously or touch on passive immunity. Active versus passive immunity are pretty much just as they sound. Active immunity develops in direct response to an infection, to your exposure to the pathogen. It can also be in direct response to a vaccine. So your body sees the pathogen, you build up a response and kill off the pathogen. So you all know about immunization. In passive immunity, you're not exposed directly to the pathogen, but you're given the benefits of it, whether it be the antibodies themselves, such as IgG crossing through the placenta or IgA in the breast milk, or even injecting antibodies that have been artificially created into a non-immune person. So somebody who doesn't carry the proper antibodies for a certain disease, you can do antibody therapy. It's not actually called that, but let's go with that. and help them to eliminate the infection. Although it is much shorter acting, of course, because once the antibodies disappear, you don't have the ability to produce more of those. Sometimes the immune system, of course, attacks itself. It can't recognize the difference between self and non-self. This is, of course, a problem because there is no treatment. The only treatment, of course, being to reduce the effectiveness of your own immune system, which then opens you to all kinds of opportunistic infections that your immune system was pushing away or pushing down, holding down. Also, transplanted tissues can often be viewed as non-self and attacked by your immune system, and hence the reason why people that have had organ or tissue transplants are on immunosuppressants to block or slow down the functioning of your immune system, and why sometimes people with transplanted tissues can potentially be harmed or die of very simple common infections. Now, blood groups are a nice way to talk about antigens and antibodies and so on, and also agglutination. So you have a certain blood type, and that blood type is defined by certain proteins on your red blood cells. Okay, we have the ABO blood groups, okay, as shown here. Blood type A, blood type B, blood type AB, which is the combination, it's codominance, and blood type O. Now, a person who has blood type A, they can receive blood type A because their immune system has been exposed to the A antigen, or the A surface protein. But if their body is exposed to the B antigen, they will attack it, and they have antibodies that are anti-B. Okay, so here you see if they get a transfusion with A, there is no reaction. But if they get a transfusion with B or with AB, they do get a reaction. Okay, their immune system attacks it. Now, blood type O, as you'll see here, this is all negative because blood type O is a naked cell. They don't carry any surface markers, any surface proteins, and as a result, there is no antigen for the immune system to recognize and attack, and hence why O is the universal donor. Now, there's another protein on the surface. There's actually more than 20 different groups of proteins on the surface of red blood cells. Typically, we only focus on two because those are the major ones that cause the biggest problems. But when doing a transfusion or transplants, they check many, many more than just those major two. The two I'm speaking of are the ABO blood group and also the Rh antigen. So some of you might know that you are O positive or AB negative. That positive or negative means you have the Rh factor, positive, or you don't have the Rh factor, negative. This becomes a problem when a mother is pregnant, since baby and mother's blood are exchanged in part. Sometimes a baby can be Rh positive and a mother can be Rh negative. Okay? I'll leave you to think about the genetics as to how that could happen. The Rh factor is a monogenic trait where positive is dominant. The Rh factor is dominant. So here, if the baby is Rh positive, the mother's immune system will attack the blood of the baby and try to kill it off. And the baby can be spontaneously aborted potentially. Now, one weird thing about this, does it say it here? No. One weird thing about this is that this is only the case in second or successive pregnancies. In the first pregnancy, if a baby is Rh positive and a mother is Rh negative, the baby is not attacked by the immune system. It's almost as if the mother's immune system doesn't really know what's going on and hasn't primed itself yet to attack the Rh positive antigen. But after having a baby that's Rh positive, this mother, if a second baby is positive, will attack that second baby and has to be on immunosuppressants during that second pregnancy or third pregnancy or what have you. Why do certain tissues get rejected? It's because of MHC molecules. If the donor and recipient MHC tissue types are well matched, then you'll have successful transplantation and still give immunosuppressive drugs. If they don't match well, then you don't use that tissue type for the transplant. We've already talked briefly about allergies. IgE antibodies are produced after exposure to the allergen. So usually, if you're allergic to something, you'll be exposed the first time, say the first bee sting, and you don't get an allergic response. But now your immune system has been primed to that to the antigen, and the next time you're exposed, the next bee sting, you'll produce a very serious response. And that's because IgE antibodies attach to or bind to receptors on mast cells, and those mast cells then will release tons of histamine, which cause inflammation. And remember, inflammation is triggered with vascular changes and so on. So here's the allergen, binds to IgE on the mast cells. IgE on the mast cells trigger release of histamine, and then the histamine leads to these allergy symptoms: flushing, red, itchiness, and so on. We've already talked about autoimmune diseases. Some examples include arthritis. There's a couple types of arthritis. That's not so important, but in rheumatoid arthritis, that's the autoimmune form, not the overuse form. The body attacks the cartilage in the joints, and as a result, you start to get bone on bone in your fingers as you use them, and inflammation occurs, and you get these big welts of scar tissue and so on inside your joints. Very painful, very damaging. Other ones include lupus, MS, and also type 1 diabetes are other examples. HIV, just to give you some idea of how HIV and AIDS are tied to the immune system. AIDS is when you have lost or are very low in your white blood cell count, specifically helper T cells, because helper T cells are infected by the HIV virus. And when the HIV virus enters the lytic cycle and starts to kill off helper T cells, your helper T cell count goes down. Remember, that's the operator in the system, and it can't communicate to B cells and cytotoxic T cells. And AIDS is the actual now full-blown disease. And people with AIDS will die of something like the common cold or the flu because they can't counteract it. They can't identify it. So results by infection from HIV, loss of helper T cells, as I just mentioned. And we're done.